Hello there! Welcome to this week's biology video. Why don't siblings always look exactly alike? Well, meiosis plays a big role in why siblings, even siblings with the same parents, don't get the same genetic information from them. So this week we're going to be learning about meiosis, but a term first that you need to know: gametes. Gametes are the egg and sperm cells, or our sex cells. And we talked about last week how those egg and sperm cells are haploid. Now, haploid meaning they only have a single set of chromosomes, so they have 23 chromosomes. Now, meiosis, what we're going to learn about is how those haploid gametes are produced. So last semester we had learned about mitosis, and that's how body cells divide or reproduce. So like skin cells, hair cells, all those are reproducing through mitosis. But meiosis, on the other hand, is how those gametes, the egg and sperm, are produced. So in mitosis, we go from one diploid cell to two diploid cells that are identical. But in meiosis, we're going to take one diploid cell and it's going to divide twice to make 4 different haploid cells. So meiosis will begin after interphase here. And so in the first round of division, our homologous chromosomes are separated. And then in the second round of division, the sister chromatids are separated. And so we end up with our 4 haploid cells. So let's start with looking at the first half of meiosis, meiosis 1, okay? So it follows that same PMAT as mitosis. So you can remember our hand motions, and you would just do them twice. So we have prophase 1, and there's something really important that happens in prophase 1. It's called crossing over. Now, crossing over, that is when the homologous chromosomes line up next to each other, and they come next to each other, they can actually cross over bits of that DNA to each other, which just leads to more genetic variety in the gametes, in the eggs or the sperm. So that's prophase 1. Then we move to metaphase 1. Remember, M, middle. Okay, but in this case, instead of just having one chromosome, these pairs of homologous chromosomes are lined up on the middle. That's how we know that this is meiosis 1, because the homologous chromosomes are still together. Anaphase, they get pulled apart. Okay, so the homologous chromosomes are separated. Telophase 1, we're starting to have these new nuclei, and then cytokinesis would split these, allowing us to move to meiosis 2, the second round of division. So in that, we still have our 4 PMAT phases. We start with prophase 2. We move into metaphase 2. The chromosomes line up along the middle. Now we have 2 cells in each of these pictures because the cells already split once. Now notice in metaphase 2, there are only single chromosomes lined up here. That's because our homologous chromosomes have already been separated. And then during anaphase 2, now the sister chromatids, the 2 halves of that, are separated. Then we move into telophase 2. New nucleus starts to form in each new cell. And then cytokinesis would split these and form our 4 haploid gametes. All right, so that was meiosis, and don't worry, you're going to get a lot of chances to practice because we're going to be covering meiosis all week this week. See you guys!